Good day, everyone. I'm Mr. Ong Wei Sian from Sriputra Sixth Form College. Today, we would like to talk about autotrophic nutrition. At the end of this lesson, you should be able to classify autotroph into photoautotroph and chemoautotroph. And you are also to describe photosynthetic pigments, explain the absorption spectrum and action spectrum of photosynthetic pigments. Autotroph synthesize organic compounds from inorganic precursors such as carbon dioxide, water, and nitrate using energy from an abiotic source. There are two types of autotroph. First, photoautotroph use light as their source of energy. For example, photosynthetic plants, algae, and certain prokaryotes. Whereas chemoautotrophs obtain energy by means of inorganic oxidation reaction, which is not from light. And all the chemoautotrophs are prokaryotic. The photoautotroph use light energy to synthesize ATP and reduce NADP, which temporarily hold the chemical energy. Then, this chemical energy of the ATP and reduced NADP will drive the carbon fixations, in which the organic molecules such as glucose are synthesized from carbon dioxide and water. Photosynthetic plants have chloroplasts in certain cells for photosynthesis. In the chloroplast, they have photosynthetic pigments such as chlorophyll A, chlorophyll B, and carotenoids they found in the chloroplast. Algal cells have chloroplast and the same photosynthetic pigment as green plants. Cyanobacteria have photosynthetic pigments, which is only chlorophyll A in the enfolded plasma membrane. Photosynthetic bacteria trap the light energy in bacterial chlorophyll during photosynthesis. Photosynthetic plants, algae, and cyanobacteria use water as the source of hydrogen for reductions of carbon dioxide into glucose, whereas photosynthetic bacteria such as the purple sulfur bacteria use hydrogen sulfide as the source of hydrogen. Then the purple non-sulfur bacteria use organic compounds such as acetate as the source of hydrogen. The other ones, which is the brown or red bacteria, they use simple organic compounds such as ethanoic acid as the source of hydrogens. Chemoautotroph. The chemosynthetic bacteria synthesize complex organic compounds from carbon dioxide and water, but they obtain the energy from other chemical reactions that they are able to catalyze, not using the light energy as in the photoautotroph. Nitrifying bacteria, for example, Nitrosomonas, Nitrobacter, they use the energy from the exergonic chemical reaction of the nitrogen cycle in order to drive the synthesis of carbohydrates such as glucose. Iron bacteria use energy released from the oxidation of iron ions to fix the carbon dioxide and water into carbohydrates. So these are how the autotroph, photoautotroph, and chemoautotroph, they carry out the reductions of carbon. Now we are focusing on the photosynthesis, that means photoautotroph. So all the photoautotroph, they contain photosynthetic pigments. Then we are focusing on the photosynthetic pigments in eukaryotic cells. So they are found in the thylakoid membrane in the chloroplast. So they are chlorophyll and carotenoids. So chlorophyll, they have the chlorophyll A and the chlorophyll B. Some seaweed, they have chlorophyll C as well. So the chlorophyll A and chlorophyll B are the main pigments of the photosynthesis, which can absorb light primarily in the blue and red region of the visible spectrum. Whereas carotenoids, for example, carotenes and xanthophylls, are the accessory photosynthetic pigments. A chlorophyll molecule has two main parts, a complex ring and a long side chain. The ring structure, called the porphyrin ring, is composed of carbon and nitrogen atoms. So this ring is used to absorb the light energy. And at the center of the ring of the chlorophyll is a magnesium atom. So the chlorophyll molecule also contains a long hydrocarbon side chains that make the molecules extremely non-polar. So this hydrophobic tail or this hydrophobic side chains anchors the chlorophylls to the thylakoid membrane. So from here you can see this is the structure of a chlorophyll. So the difference between chlorophyll A and chlorophyll B is at this part, the functional group, whereas the chlorophyll A has a metal CH3, and for chlorophyll B is a carbonyl CHO in the position. So this is the magnesium atoms in the center of the porphyrin ring. So this is the hydrocarbon tail. Okay, so this difference, that means the functional group difference, shift the wavelength of the light absorbed by the chlorophyll A and chlorophyll B. So chlorophyll A appears blue-green, and whereas the chlorophyll B appears yellow-green. Carotenoid. Carotenoid consists of the carbon rings to a change of 18 carbon atom here. Okay, so the carotenoid examples of the carotenoids are carotene and xanthophyll. So the difference of the carotene and the xanthophylls are the presence of hydroxyl group at the bottom here. Okay. Chlorophyll is the main photosynthetic pigment, and chlorophyll A is the pigments that initiate the light-dependent reactions of the photosynthesis. So in the coming lessons, you will study the light-dependent reaction, and then you will see, you will study the role of the chlorophyll A. Chlorophyll B and the carotenoids, they are accessory pigments that participate in the photosynthesis, and they also protect the chlorophyll A especially. Okay, from the excess light energy, they could easily damage the photosynthetic components. So these are the summary of the pigments of photosynthesis. So the major pigment, which is the chlorophyll A, the color of the pigment shown is blue-green, and they found in plants, algae, and cyanobacteria. Whereas chlorophyll B and the carotenoids, they are accessory pigments, and they're found in the plant green algae, okay, in the chlorophyll B, and the carotenoids found in the plant, algae, bacteria, and archaebacteria. Visible light. Visible light represents a very small portion of the vast continuous range of radiation called the spectrum, called the electromagnetic spectrum. So from the electromagnetic spectrum here is they have a long wavelength. This is a short wavelength. So the short wavelength is more dangerous. And then this in the middle here, the average here is the visible light. Yeah, visible light. So from 380 nanometer, which is the violet color, to 760 nanometer, which is the red color. And after that, here is a far red. The light is composed of a small discrete particles of energy called photons. Okay, if you study physics, the light is called a photon. They have a small discrete particles, which is a contact energy. And this one is known as the photons. Then the energy of a photon is inversely proportional to each wavelength. That means the shorter the wavelength of light, the higher the energy of the photon. So that means the gamma, okay, the gamma ray has higher energy compared to the UV ray. So when a molecule absorbs a photon of light energy, okay, let's say this atom absorb a photon of light energy. So one of each electrons becomes photoactivated. Remember, we are using the terms photoactivated. So these photoactivated electrons, they will shift from a low energy atomic orbital to a high energy orbital, atomic orbital here. Okay. So which is distant from the nucleus. Then there are two possibilities. Okay. The energetic photon is emitted when the electron returns to the low energy orbital. Okay, so these photoactivated electrons now is here. So if these photoactivated electrons returns to the low energy orbital, so an energetic photon is released. Or that is the other way, which is the photoactivated electrons is accepted by an electron acceptor, which becomes in the reduced process. That means this electron acceptor accept the photoactivated electrons becomes reduced. So this electron acceptor carries a high energetic electrons. This one happens in the photosynthesis. In photosynthesis, some photosynthetic pigments undergoes this process, which release the energy to the chlorophyll A, in which the electrons, the photoactivated electrons in specialized chlorophyll A will be photoactivated and received by electron acceptor. So we will further discuss these photoactivations in light-dependent reactions. So for the photosynthetic pigment, we can show the absorption spectrum. That means which spectrum of the wavelength of light is most absorbed by a particular photosynthetic pigment. So this is known as absorption spectrum of a pigment. So it's a plot of a pigment, that means photosynthetic pigment, absorption of light of different wavelength. So here is different wavelength of the light. Then here they show you the different what we call the photosynthetic pigments that can absorb the different wavelength of light. Okay, so each pigment has a characteristic absorption spectrum. For example, they can look at the chlorophyll A. So the chlorophyll A, they absorb most of the wavelength of light in here, compared to the chlorophyll B, which is almost here, and the carotenoid is around here. Then you can see there is no or less absorption of the light wavelength in the green color, the green region. So chlorophyll A absorbs light, okay, of 430 nanometer and also the 662 nanometer here, blue, violet, and the red color. Whereas the chlorophyll B absorbs light of 453 blue and 642, okay, and the 642 here. And the beta carotene, okay, beta carotene absorb light of 450 nanometer violet and the 475 nanometer, which is the blue green. Then you can see that neither of these pigments absorb the photons in this 500 to 600 nanometer, which is the green region. So that's why the green light is reflected into our eyes and the leaf appear green color. In contrast, action spectrum of photosynthesis is a graph of the relative effectiveness of different wavelengths of light in promoting photosynthesis. That means it is the average, okay, or the sum of the what we call the absorption of light, okay, for all the photosynthetic pigments. So a peak shown in an action spectrum corresponds to a maximum absorption. So for example here, the maximum absorption is occurred in this region and this region. And again, in the region of green, there is the least absorption. So this graph is the action spectrum of photosynthesis. So which is different to the absorption spectrum of photosynthetic pigments. This one is referring to one or a particular photosynthetic pigment, and this one is the action spectrum for photosynthesis. So the action spectrum of the photosynthesis actually is obtained by the German biologist, okay, Engelmann. Okay, first they use the spiral gyre, okay, okay, and then they have a prisms. So the light is passed through the prisms, so that's why they come up with the rainbow color. So then they use the aerobic bacteria and look at and study the accumulations of the bacteria along this different color of light wavelength. Because during the photosynthesis, the spiral gyre produce oxygen. Then the aerobic bacteria will accumulate at the region where there is high concentration of oxygen. Then you can see the green region here has the least bacteria, the aerobic bacteria accumulate. So that means less photosynthesis taken place in this green region or the green light. So from this, the accumulation of the aerobic bacteria, they can reflect the action spectrum, even though it is not the most accurate one. But at least this experiment show that the basic, okay, they give the opportunity for scientists or biologists to investigate further. Then this is the checklist we go through this lesson, this video. I hope that you are able to classify the autotroph into the photoautotroph and chemoautotroph, and you are able to describe the photosynthetic pigments, the chlorophyll, chlorophyll A, chlorophyll B, and the carotenoids. And you are able to explain what is absorption spectrum of the photosynthetic pigments and what is action spectrum of photosynthesis. So the next lessons we will talk about the light-dependent reactions. That's all for today's lesson, and thank you. Bye.